Good afternoon, everybody. Good afternoon. Good afternoon. Thank you for coming. This is a much larger crowd than I expected, actually, on a beautiful sunny day on Chestnut Hill College. Well, good afternoon. It's a pleasure to have you present and to welcome back our series speaker for the second time. This is the first time ever that we've had a returning speaker to this biomedical seminar series. I'd like to acknowledge all of you that are present here today, including Dr. Andra Basu, Vice President of Academic Affairs and Dean of the Faculty, our current faculty members, staff, students, and also any guests that are with us today. Michael obviously is no stranger to Chestnut Hill College, as many of you know, and who would be with Dr. Lakshmi Atchison, his beloved wife, and a faculty member here at CHC for over 3 decades. This lecture series was actually the brainchild of Lakshmi, and it was her baby for 3 decades. She invited only the brightest and best of the professionals who stood at this very podium presenting their groundbreaking efforts in science and medicine, including 3 Nobel laureates. Michael, your accomplishments, though, certainly rank well amongst those invitees that have graced this podium. It's also fitting on the centennial celebration of Chestnut Hill College to welcome Michael and Lakshmi back home. So welcome home, Mike and Lakshmi. Mike, it's such an honor to have you discuss your exciting and important research with us today and to inform the audience of other remarkable achievements in your life and talents, which are many, that go even farther beyond the silo of science. So let me introduce Michael. Michael got his BS degree at SUNY Albany, I believe, PhD at NYU School of Medicine. You are currently a professor in the Department of Veterinary Sciences, University School of Pennsylvania Veterinary Medicine. You are director of the VMD PhD program. That's a program for trained veterinary scientists in medicine. You are also director of the NIH Boehringer Ingelheim Summer Research Program for veterinary students. I guess that gives them opportunities for summer internships to train vets in their profession, which is a wonderful thing. So there's 3 aspects of Michael I'd like to talk about today. And the first one is Michael as the scientist, okay? Michael, one recommendation is when you have a group photo, maybe better background, I don't know. The letter, the mailroom is not the best. Anyway, I really love this picture to the left because it shows 2 former CHC alums, very significant students that graduated from this institution. Top left is Dr. Aisha Gaius. She is currently at NYU, a pulmonary critical care physician. And on bottom left, well, one of the first males that joined us when we went co-ed, by the way, who's now Dr. Brandon Youngman. He's a psychiatrist. He's working at the VA, serving veterans that have obviously mental disorders. Next slide. So let me run through some of Michael's credentials. I just mentioned he's professor of biochemistry at Penn. He is a great teacher, obviously won the Lindback Award for Excellence, and also UPenn's Dean's Award for Leadership in Basic Science Education. He's been a prolific author, publishing over 85 peer-reviewed publications, and I mean in very significant journals. He has had continuous grant support from numerous agencies, including the NIH and all of these other foundations that you see listed there. And remarkably, he's trained over 70 postdoctoral scientists, graduate students, undergraduate students, the 2 that I just mentioned that were graduates from our institution, and veterinary students, obviously, and even high school students. Last fall, Penn put out a news release from the Perelman School of Medicine, and it hit the airwaves, so to speak, and it was ranked as within the 5 top % of research that was flagged as being very significant in the field. So it's quite an accomplishment. That title of that paper that triggered all this is "YY1 Knockout in Pro B Cells Impairs lineage commitment enabling unusual hemopoietic lineage plasticity. I got through that. But he'll explain that too. It's very significant work. It has a lot to do with altering stem cell characteristics. And now for something completely different. So Michael is obviously a wonderful scientist, but he has two other significant achievements and talents in his life. The first one is he's executive director of a foundation that he started, Helping India Together. So it's a foundation that aids in education for underprivileged children, also helps marginalized women. I think you also have a nursing program, training nurses, because there's critical care needs in the country of India. And I also see that you have a great concern for the environment. So some of these funds that you acquire through your organization go towards that effort as well. So you are a humanitarian. And finally, I will end with this. Michael is actually a rock star musician. So he actually plays the guitar very well. And this will be the last one. I hope it works. There it goes. Here we go.
If you don't let your hair down, woman, can't have no fun at all.
So it's a pleasure for me to speak at Chestnut Hill College. I've been here so many times over so many years, and it's an honor to speak in the Biomedical Seminar Series. Yeah, I am a repeat offender, but the least impressive of the list of Nobel laureates, division heads and so forth. It's really an incredible honor to speak in this series. Today I'm going to be talking a little bit about our recent work in regenerative medicine, a field that is actually relatively new to me. I study how genes are regulated in the immune system, but currently we're doing work related to that, but more leaning towards regenerative medicine. And so lineage commitment is, when does, say, a liver cell become a liver cell? That's what it's really about. And so all cells in your body come from a single cell, right? The fertilized egg. And then you have cell divisions, does the mouse on this, does this show up? No, it doesn't. Am I able to use them? Okay, I'm not. So on the far left, fertilized egg, and then you go into 2 cells, 4 cells, and so forth. And eventually you get, a funny looking squiggle of cells on the far right. And then ultimately out pops out a mouse or a human being, and a human being with, and the mouse with organs, right? Lungs and stomach and liver and all that. But all that came from a single cell. So when does a single cell change into a differentiated cell, like a liver cell? Well, you're made up of billions of cells in your body. You started as a single cell, and that ultimately made all the billions of different cells. And as those cells divide and multiply and you increase the number of cells, some of them differentiate into different cell types, heart, liver, and so forth. And there is a position in development in all the cells in your body where they actually became committed to become a single cell type, right? When that liver cell is going to be a liver cell and not going to become anything else, or your heart cells are going to be heart cells and they're not going to change into anything else. And so that's the time when cells are committed. And those committed cells express specific genes. They make proteins that make a heart cell do what heart cells do, and they contract and, pump blood throughout your body. So what controls that are transcription factors, genes that that turn on and off specific genes. Now, I said that a heart cell stays a heart cell and a liver cell, but you can actually change those cells by putting different genes into them, and that's what this slide shows. On the far left is a fibroblast cell that's turned into a muscle cell. And that was done because a gene called MyoD was put in those cells. MyoD is a transcription factor, a gene that regulates cell development. And it turns on the muscle cell program, so those cells now are muscle cells. And there's other examples there, like fibroblasts turning into beta islet cells in your pancreas, cardiomyocytes, neurons, and so forth. So you can actually change cells, but you're doing it by putting something into them. These names across the slide stand for certain transcription factors, these regulatory proteins that turn on genes needed for those cell types. And the third panel from the left, you can actually make cells go backwards. So you can make a fibroblast, which is sitting in your tissues, and turn it into a pluripotent stem cell that can then become almost every other cell type. But again, in that case, you're putting specific genes experimentally into those cells and turning them into something else. Now, this third panel from the left of making a pluripotent stem cell is really the bedrock right now of regenerative medicine. And in regenerative medicine, people oftentimes take these cells, send them back in time to an earlier state, this pluripotent state, where they can become many cells, and then they manipulate those cells in some way to make another tissue, lung tissue, adrenal tissue, and so forth. But all these depend on putting those regulatory proteins in, and I'm going to describe something where you maybe don't need to do that, and yet you can turn cells into other types of cells. So my lab studies B cell development. Probably have heard of B cells. They're lymphocytes that make antibodies that protect you from infections. So when your cells are making antibodies, those are B cells. And so I look at B cell development, and this slide shows all the different lineages in the hematopoietic system. It splits into two basic branches, and the left side makes cells that you've probably heard of as well, things like basophils and eosinophils and monocytes, et cetera. And on the right side are things like B cells and T cells that protect you from foreign antigens, viruses and bacteria and every bad thing that you don't want attacking your body. So my lab looks at B cell development, and this slide shows sort of the linear pattern of going from a very early cell, a hematopoietic stem cell, that then turns into a B cell in the end. And as the cells move along that pathway, there's a period of time when they can't go back. And that's where that arrow is. At the pro-B cell stage, the cells are committed to only be pro-B cell or B cell lineage cells. They can't go backwards. So that's the committed step. Now, how do these, genes get turned on to commit a cell? Well, genes are turned on and off. And the top panel on this shows TF stands for a transcription factor, these proteins that regulate gene expression. And they bind next to the gene in DNA at sequences called promoters, or they bind at DNA sequences called enhancers. And those proteins turn on the right genes in a particular cell for that cell. So a liver cell will have transcription factors that turn on liver genes. And then there's repressor proteins in the bottom panel, the red ovals, that shut genes off. So if you're making a liver cell, you don't want to make things that a heart cell makes. And so those genes are shut off, okay? And I'm not going to get into the weeds of how that happens, but there's two things going on. When your cells become a particular cell type, certain genes are on and other genes are shut off. And that's what makes them that cell. Now, one protein long ago was discovered that actually did both. It activated and repressed transcription. And in those days, that was considered unusual, and it was given the name YY1 for yin yang 1, the Asian symbol yin yang 1. And YY1 was found that it could activate and repress transcription. It was sort of made a little bit of a splash because nobody expected that. Now we know that that's actually not very unusual. And so the way YY1 does this to turn on genes is it makes loops between DNA regulatory sequences to turn on a gene, and it recruits repressor proteins to shut off the other genes. So it does both things. Now, YY1 has a history that I'm quite acquainted with because I came to Philadelphia to do a postdoc. And after my postdoc, I took a position at the University of Pennsylvania in 1989. And the very first thing that my lab did when I was a young punk assistant professor was we cloned YY1. And so I was in YY1 from the beginning, but that's not what my lab first called it. So a graduate student in my lab named Kyung-Suk Park cloned YY1 in my lab, but we called it NFE1 because it bound to a DNA sequence called E1. It's really dull, right? Crappy name. Simultaneously, here in Philadelphia, Fox Chase Cancer Center, Bob Perry, who was actually my postdoctoral mentor, had a postdoc in his lab named Harry Haraharan, and he cloned the same transcription factor, and he called it Delta because it bound to a DNA sequence that Bob had named Delta. Again, a crappy name. Meanwhile, a friend of mine, Yang Shi, in Tom Schenk's lab at Princeton, cloned YY1, and he called it YY1. Now, that's because YY1 is a cool name, right? That's a cool name. And so if you're going to clone something, make it a cool name. If you're going to name something, make it a cool name. Don't do NFE1, don't do Delta. Make it a cool name. And so I actually learned that lesson. I learned a domain in YY1, and I called it the repo domain. Now, it stands for repressor polycomb. That's repo, but I don't know how many old timers here know what a repo man is. Yeah. So that's the guy that repossesses your car when you've not made your payments. And there's a cult movie called The Repo Man, right? So I learned my lesson. If you're going to name something, make it a cool name. So Yang Shi, the guy that named it YY1, actually was the one that found out that if you knock out YY1 in B cells, they stop development at the pro-B cell stage. They don't go any further. They just sit there. And that is the committed cell. You know, pro-B cells are not going to become anything else, but they can't go forward to become B cells either. So they're sort of frozen in time. And so there was a postdoc in my lab named Anupam Banerjee who said, I wonder what happens in pro-B cells when YY1 is knocked out. So he looked at gene expression profiles, and I won't bore you with these profiles, but what he found was some of the B cell genes weren't expressed as high as they used to be, and some genes for other lineages, like T cells, macrophages, neutrophils, and so forth, some of those genes increased in expression. So what Anupam said was maybe YY1 knockout pro-B cells will no longer be committed, but will turn into T cells. And so that was his idea. You know, he wanted to try that. My response was, that's crazy talk. That will never, ever happen. But go ahead, knock yourself. You're wasting your time, right? That was my response. But it worked out beautifully. So if somebody has a crazy idea, let them do the crazy idea, because sometimes it works. So there's reasons why things come out differently than you expected. The most common is you actually screwed up the experiment. Okay, that's the most common problem. Sometimes your idea is wrong, you get a different result because you thought one idea, but it's not really true, and the experiment comes out different. And so that can happen. And sometimes something really, really incredible and cool is happening. And that's what I want to convince you is happening with this YY1 knockout, that something really cool is happening. So what Anupam did to make them into T cells is he took B cells out of a mouse, okay, pro-B cells. He purified them out of a normal mouse and a YY1 knockout mouse. And he put those cells, independently onto cells that push cells towards T cells, T lymphocytes. And then he looked, did they become T cells or not? Or did they just sit there? Now the cells, and this is a piece of data that you don't need to know what any of this means, but on the left side, there's a rectangle inside the box, or a box inside the box. That's empty. And that's because normal, and that's where T cells should lie, and that's where the normal pro-B cells went. Nothing. They didn't turn into T cells. And then the panel below that has a blotch of 90% of the cells are in that box. And those were the YY1 knockout pro-B cells that turned into T cells. And the graph on the right side just counts that. The left, 0 cells, that's the normal pro-B cells. The right tall column are the knockout pro-B cells that all became T cells, or at least 92% of them. And there's controls in here to prove that those really were pro-B cells and weren't contaminants. They were real pro-B cells, YY1 knockout pro-B cells that turned into T cells. And so after Anupam left, 2 people took over this project, Sharmistha Banerjee and Suchita Hodwadkar. And Suchita's been in my lab since 2002, so she's a lifer. Sharmistha was a postdoc. She was in the lab a few years. And they showed that these cells not only had some B cell characteristic, but they also looked a little bit like T cells. But we wondered, are these really T cells? We changed from one cell type into another. It's like going from a liver to a stomach. Well, those are very different cells. How are you going to tell if they're really different? You look at the genes that are expressed in those cells. And so 3 people worked on that in my labs, Sulagna, Sarah, and Nazreen. And they're currently in my lab working on this project. And this slide is terrible. You can't see the data, but at the very bottom are Venn diagrams. And what they're showing is that the pro-B cells that we started with, the YY1 knockout pro-B cells that we think turned into T cells, when you look at the gene expression patterns, they're 90% identical. So that is incredibly high. They really look like T cells. And the left Venn diagram says that comparing our T cells that started as pro-B cells to normal pro-B cells, there's only 10% similarity. So it's 90-10. They're 90% looking identical to a T cell with a 10% hangover of some B cell gene still being expressed. So how does that work? Was it like they just turned on and off, or was it a gradual thing? And I was thinking that there's probably a gradual transition from the B to T cell phenotype. And so we decided to do a special type of sequencing called single cell RNA sequencing, where we could determine the actual sequence in every single cell instead of looking at an average. And when we did that, we found something very, very surprising. And so this is a little difficult to describe, but if you look at the top panel, the second panel, each dot in these panels is a cell, and the color of that dot tells you what type of a cell it is. And so in the top panel B, the second one, those are YY1 knockout pro-B cells before they went on to feeder cells to make them into T cells. And they look like B cells, right? That color is the color of B cells in this program. The left panel there are the ones that were differentiated into T cells, but caught halfway before they got there, when only about 5% of the cells were T cells, and it looks like the Wild West. Every cell type has shown up. It is not a T cell that showed up. It is 5% T cells, and then everything else. And the big panel C points out what the names of those cells are. So there's monocytes in there, dendritic cells, macrophages, neutrophils, you name it. Every hematopoietic cell type is in there, including stem cells, which is a little bit unusual. It implies the cells went backwards. all the B cells are gone, even though there's a little part there that's labeled as B cells. And I looked into the program to see how many B cells really are there. It turned out to be 33 B cells out of 16,000 cells. So there's hardly any there. They all disappeared. And this is a graph that just shows that. And this is single cell RNA sequence, the top panel of a B cell gene. The first panel is the cells that have been differentiated. They've lost expression, they're blank. And the right panel is the B cell pattern. I wish this pointer actually worked. Or does it?
Yeah, it does.
It doesn't show up on TVs. Never mind. All this panel is showing you is that the B cell pattern completely disappeared. There's no B cell genes expressed, but monocyte genes are expressed, dendritic genes, T cell genes, and even stem cell precursor genes are being expressed. It's like every lineage pathway went on. This is like the whole differentiation process takes 3 weeks. We caught it right in the middle. At 2 weeks, it doesn't look half B, half T. It looks like I said, the Wild West. Every gene is gone on, and there's clusters that represent different cell types. If you wait the full 3 weeks, everything turns into a T cell. So we caught it midstream, seeing what I did not expect. I expected a gradual change, B to T, and instead, every gene on Earth seemed to go on. Not expected. We did not anticipate that result. And what we've determined since then, forget the data on the left, that just proves that this is happening in mice. So everything I showed you to now happened in a Petri dish. Cells we put in a Petri dish. This is actually data from mice, okay? Taking cells out of the mice and looking, and we can see B cells that became monocytes, that became dendritic cells, and even became hematopoietic stem cells. And if they became hematopoietic stem cells, they can go into any cell type in the hematopoietic system. So I'm assuming every other cell we look at, we're looking at that now, and every other cell type in the hematopoietic system will probably have cells derived from YY1 knockout pro-B cells. Again, did not see that coming. But my fantasy is this isn't peculiar to B cells in the hematopoietic system. My fantasy is that it's actually applicable to every cell type, that these pro-B cells can become anything, including cardiac cells, neural cells, intestinal cells, you name it. My hypothesis is that's what they're going to become, and we have a small amount of data that actually says that may very well be true. So we use some very high-power molecular techniques, ATAC-seq, single-cell ATAC-seq, Hi-C, that measure things going on in the genome. Okay, and I'm not going to show you that actual data because you know it would make your mind numb. But to me, it's like the coolest stuff I've ever seen. So, you know, my life isn't so exciting. This was cool. When we saw this data, it's like, now we actually see why this might be working. So I'll just sort of lay out what's happened. So transcription factor binding sites are all over near these genes. Every gene has where transcription factors bind to turn that gene on or off. And we found that those transcription binding sites that regulated B cell genes were no longer as accessible when we knocked out YY1. So if you knock out YY1, all of a sudden the B cell genes, they're not as accessible, they're not as well expressed. Meanwhile, genes to other lineages started being more accessible. The transcription factors that bind to those genes became more accessible. And then we looked at the genes themselves, and the same thing. The B cell genes were not as accessible, whereas the alternative lineage genes were accessible, meaning they could be turned on. And we looked at the DNA loops that regulate gene expression, and the B cell loops fell in number at B cell genes, while the loops at other genes increased. New loops appeared, meaning they were ready to be expressed. So if you knock out YY1, you get these genomic changes within the cell that reduce the commitment of the cell it is and prepare the cell to become something else. They're not going to become something else unless you push them, but they're ready. So YY1 is able to confer stable expression on the B cell lineage in B cells, and if you knock it out, then it's no longer as stable, and other genes that were stably off are now ready to go on if you put them in the right environment. And so how is this working? Again, a summary of data that you don't need to know the specifics, but I've told you that YY1's a transcription factor, and there are many factors that bind to genes and turn them on or off. So the first thought was that YY1 is binding to all these alternative lineage genes and either turning them on or off. And it turned out it was binding to none of them, 0. It wasn't binding to the B cell transcription factor genes or any other lineage. So that's a little bit of a surprise. Instead, again, data, but I'll just tell you what it means. Instead, YY1 was regulating more general things. When genes go, when transcription factors bind to genes, they recruit general proteins that either make the gene more accessible or less accessible. YY1 was regulating all those general activators and repressors. So when you knock out YY1, the activators, the co-activators and repressors went down in expression. So you have a case where, yeah, the genes are sort of on, but they're not quite as strongly on because you're infecting the general stuff, not the tissue-specific stuff. I don't know if this is making sense to you, but when you sort of put it all together, it does. Basically, on the left is a normal gene being regulated by YY1 in a B cell, okay? And the blue, green, and orange are these positive acting co-activators that keep that gene on. When you knock out YY1 on the right panel, well, YY1's not there, so the loops fall apart, but also those co-activators are down in expression, so you have fewer of those to help keep the gene on, so the gene falls. It stops, it goes down in expression. At the same time, YY1 represses genes. Remember, it's yin-yang 1, it does both. It represses genes, and it does that by recruiting co-repressors. And so when YY1's there, the co-repressors and the repressive proteins that make chromatin structure silent are there, and the gene is off. But on the right panel, you lose YY1, co-repressors, and the genes that keep genes, or the proteins that keep genes off reduce in expression, and so all of a sudden you have them going up in expression. So it's doing both of these things, and it's by a general mechanism, not hitting the specific transcription factors that regulate each lineage. And that's why I think it can be a universal mechanism. So if you were going to use this for regenerative medicine, you would first, any cell type, take any cell, a neuron, a heart cell, muscle cell, intestinal cell. There are ways to knock down gene expression transiently. There's like oligos, RNAi that you can use. There's a number of ways. You can use CRISPR, but that's generally a permanent deletion. But there's ways to transiently knock down gene expression. So you take whatever cell you're interested, transiently knock down YY1, and the lineage-specific genes will be reduced, and the alternative lineage genes might increase a little bit. Then you put the cells into an environment that stimulates development of the lineage you're interested in. And there are in vitro mechanisms to do this. You can make neural cells, you can make liver cells, there's all sorts of cells you can make. It just so happened we used a system that made T cells, right? And that's the system we use. But now we're looking in these other systems. So currently we're looking at neural cells, intestinal cells, and adrenal cells, because I've got collaborators that know those systems well, and they're providing us the reagents. So you put them in that environment, and the B cell genes are now going to be repressed because there's nothing to keep those B cell genes on, and the alternative lineage genes are going to be stimulated because that's what the in vitro system is designed to do. And you're gonna, in my fantasy world, they're gonna turn into that cell type. All right, but that's transient, right? You've got to remove the block to re-express YY1 that now locks on the new correct genes and off the alternative lineage genes. So you basically change from a pro-B cell into whatever lineage your in vitro system has developed. And so my fantasy is this is a really non-invasive way. You're not putting transcription factors in like, slide 20 slides ago where there were all these cells turning into other cells. You were putting a transcription factor gene in. Well, the problem is you put something in the cell. In this case, we're actually not putting anything in the cell other than these molecules that will transiently knock down a gene and they degrade. They're then gone and gone forever. And so my fantasy is it's a relatively safe way to apply this to regenerative medicine. And so these are things that we're doing now in the lab, and we're pretty excited about it. So can it be universal? Yeah, I hope so. The early indications are pretty strong at the moment, and I think it's going to work. But you don't know until you know. That's why you got to do the experiment. I hate that. If only I could do things on ideas. You can come up with a million of them, but in science you got to do the research, do the experiments, do them multiple ways to prove that what you think you've found. And so that's what we're at the moment. These are the folks that have been involved in the work. Suchita, as I mentioned, has been with me for 20-something years. Sulagna, Sarah, Nazreen, and Dawe have been working on this project the last few years. Anupam and Sharmissa at the bottom are now in new labs. And then I've got some collaborators. They're all at Penn in this slide. Dave Allman, Chris Langner, Josh Rhodes, and Sherry Wang have been collaborators that we've worked in, not on the exact data presented here, but in helping with some of it. So thanks for listening, and I hope to be able to tell you more in the future.
Any questions?
Hi, so it makes sense like in vitro that you take a pro-B cell and give it family stromal cells or whatever, they develop into T cells. But in vivo, how would you get a pro-B cell to make people in vivo?
Oh, that's a good question. So you wouldn't be able to get it to go in vivo, right? You'd have to do it in vitro and then put it in because pro-B cells stay in the bone marrow by and large. But, I shouldn't quote data that I just saw yesterday, right? But we have a mouse that will fluoresce YFP when it's activated by, a Cree driver. And we've got it in a YY1 knockout background. And it looks like there's some alternative tissues that are lighting up fluorescent. Again, one experiment, one mouse yesterday. So in theory, it shouldn't work. But, I told Anupam he was out of his mind, and yet. Yes?
So you have multi-pigmented arcs in their cells. And do you think in the future this technology, this new site, allow for, say, creation of a heart with my own DNA that you could say swallow and maybe it could my heart replace my heart?
Can't do too much about the emotional broken hearts, but right. So yeah, my fantasy is that it is applicable to that. Now, making a full organ is more complex, obviously, and it's way beyond my expertise to make actual organs. But making the cells that could seed, a synthetic structure where you would build an organ on, maybe.
And my DNA, so could come to
Could go take your cells, yeah. You would take a patient's cells, and that's the beauty of it, right? You're able to, obviously, it would probably be prohibitively expensive in today's world, but maybe someday not. You would take a patient's cells, knock down YY1, put it in a differentiation system to make whatever is needed, and then, reverse the block.
I was in this man, I see cancer, so he's waiting.
Right, so the question is about, can this be used for cancer cells? You could imagine it being useful, particularly, with like whole body radiation treatments where you pull out some cells from a patient before you do heavy duty irradiation. Like, when there's stem cell transplants, hematopoietic stem cell transplants for blood cancers, that's basically killing people and then bringing them back to life with donor cells. But presumably this could work in any tissue that you are destroying and then replacing, let's say you've got a defective, liver, let's say. You could pull out other cells, change them from, say, heart cells into liver cells and transplant after, doing a surgery to remove bad parts of the liver. In theory, it could work. Yes?
So, good for the you develop priority. So we have this church that on walking road cell lineage. You have an idea what the signals are to say, okay, why as opposed to something else?
Yeah, that is a good question, because every cell has an epigenetic landscape, right, of accessible and inaccessible. So some cells may be more amenable to manipulation to make them go into different lineages. And maybe you would go to, hematopoietic stem cells are relatively accessible, but they can't turn into other cell types. Right? They're in the hematopoietic system. They make the hematopoietic cells. But you could imagine you could take a precursor cell like that and basically erase that by knocking down YY1 and turning it into other cell types. I don't know, does that answer the question? I'm not sure.
You could ask it. I know what it was saying was intuition and you have to chip the guard.
Oh, yeah.
Well, it was why here?
Yeah, so most of the genome is inaccessible in most cell types. And so I'm amazed that this works at all because of what you raise, because the alternative lineage genes are in, chromatin that's condensed and not really generally accessible. And yet when we knock out YY1, somehow every hematopoietic cell can be made. Well, I shouldn't say it. We've shown 3 or 4 of them, but I'm assuming they all can. Yeah, why does just knocking it down allow those genes to, be now accessible? I don't know. Yeah. Yes?
You have the racing on where the stem pen
Yes.
is funny to this research to
Well, I hope so. So at Penn, there's been a handful of grants that were shut down, and they were grants making vaccines. And it's just recent. Penn was threatened with indirect cost cuts. That's like every dollar I get, the university gets added on, more money for infrastructure and all that. That cut probably won't happen because it's a contract, but when the contract is up in another 1.5 to 2 years, then those cuts seem possible. Penn just lost $175 million on the basis of a transgender argument of an athlete, a long time ago. I don't know what's going to happen. The future seems murky in terms. So a lot of universities had to rescind offers to graduate students. Penn didn't rescind any, but we reduced offers by 1/3 because just worried about not having the money. Everybody, students there are obviously worried, but the university's guaranteeing they'll be taken care of until they complete their degree. Yeah, it's a murky world, not one I anticipated. Yeah.
Just one more science question.
Yeah.
So obviously the YY1 knockout might survive. Do they get immunologic efficiency?
Okay, so yeah, I should repeat the question. So the YY1 knockout mice are viable. Do they show deficiencies? Our knockout only knocks out in the B cell lineage, and they arrested the pro B cell stage. So they don't make mature B cells. They don't make no antibodies. They're viable, because they're grown in pretty clean environments. Their T cells are fine because that lineage isn't disrupted, but the B cell lineage is, basically wiped out.
And they're susceptible to infection.
You would think so, but they seem to survive okay. Now, they're in controlled environments, with, screening all the time for pathogens, but in the wild, yeah, they would never survive. they'd be gone.
Right?
No, we don't generally. I mean, if we do some experiments, we do. If there's like a transplant, they get antibiotics, but not normally. Yeah.
Hide me.
Well,
Is YY1 expressed in a lot of the organs and tissue?
oh, forgot to mention that. It's expressed in every cell type, and that's why I think it can be universal. Sorry, I didn't mention that. It's ubiquitous. It's in every cell type, and therefore, if it works in one cell, my hope is that it works in every other cell.
So, Michael, is there any information or your any thoughts about what the YY1 may regulate some master regulated DNA installation? What's our system application?
It does regulate some HDACs.
Okay, so
And that was the slide showing some modifier genes reducing an expression. So it does do a bunch of those. I can't remember offhand the exact ones that we saw did drop.
it should, yeah, you actually see the actual stuff, changes in the operation or the stuff application.
So that's a good question. We haven't looked. I've been spending a fair amount of time looking at the ATAC-seq, single-cell ATAC-seq, traditional ATAC-seq, and Hi-C and alternative lineage genes. And it's, like coming out like I wrote it, right? That's never happens, right? It's just coming out like, well, I hope it does this. And it's like, yeah, well, that worked. So,
You look at it person expression
no,
relative to the RNA.
no, we've not looked at the protein level, only at the RNA level, right? Well,
If there's no more great questions, let's take.
thank you.
We're done.